So we'll be starting with molecular revision today. So we'll be doing the chapter molecular inheritance. Okay, so let's first understand what DNA is. We know that DNA and RNA comprise the genetic material. Often, DNA is described as a polynucleotide because it is comprised of monomers of nucleotide. It is nucleotides that together make up DNA. So monomer for DNA is nucleotide. Now nucleotide is composed of three components: a pentose sugar, a phosphate group, and a nitrogenous base. So we have the pentose sugar, we have the nitrogenous base, the nitrogenous base is attached to the pentose sugar through the first carbon with the help of a glycosidic bond. It is N-glycosidic bond. So the nitrogenous base is connected through N-glycosidic bond to the first carbon of the pentose sugar. And to the fifth carbon, phosphate group is attached. The phosphate group, there is a phosphorus atom to which 4 oxygen atoms are connected. Now due to this phosphate group, DNA is acidic as well as negatively charged. So it is due to the presence of a phosphate group that DNA is both acidic as well as negatively charged. So phosphate group is responsible for the acidic property as well as the charge of DNA. So this sugar is pentose sugar. It will have 5 carbon. The 5th carbon is projected outwards. Now this pentose sugar can be of 2 types. It can either be ribose sugar or be deoxyribose sugar. If at the 2' carbon a hydroxyl group is present, then that is the ribose sugar. And if at the 2' end only hydrogen atom is there, then that is deoxyribose sugar. Ribose sugar is present in case of RNA, whereas deoxyribose sugar is present in case of DNA. The presence of oxygen in hydroxyl groups makes RNA highly reactive. So RNA is highly reactive due to the presence of a hydroxyl group at the 2' end. Whereas in case of a deoxyribose that is present in case of DNA, we only have hydrogen atom. So we can say DNA is more stable than compared with RNA. So till now we have looked at just one bond, that is glycosidic bond, that connects the sugar with the base. So how are 2 monomers joined to each other? One monomer, that is how is one nucleotide joined with another nucleotide? So 2 nucleotides are joined to each other with the help of a phosphodiester bond. So phosphodiester bond is the bond that links two nucleotides, or I can say monomers, together to form DNA. Okay? So many such nucleotides are joined to each other with the help of phosphodiester bond. So this is phosphodiester bond that is connected all these nucleotides together, and together they are going to comprise DNA. Okay. So this, we have drawn one strand of DNA. So in the secondary structure, DNA exists as a double helix. This double helix structure of DNA was discovered by Watson and Crick. They came to this conclusion based on the X-ray diffraction given by Rosalind Franklin. Okay. So Watson and Crick said that the DNA in its double helix form exists as a secondary structure. So DNA has a double helix conformation in the as their secondary structure. So in primary structure, DNA is made of all these nucleotides that are connected to each other with the help of phosphodiester bond, whereas in case of the secondary structure, DNA exists as a double helix. Now this double helix is right-handed. In the secondary structure itself, DNA can exist in different conformations. The conformation that we have to study is bDNA, and bDNA is again a right-handed helix. Okay. It's a double alpha helix. Now this alpha helix conformation is formed by two polynucleotides. There are two polynucleotides that are participating in the double helix structure. So what is this double helix structure made of? Now each polynucleotide chain of this double helix is actually composed of sugar and phosphate group. So in the polynucleotide chain, what we can observe is the sugar and the phosphate group, they are forming the backbone. They are forming the backbone of the double helix structure, whereas the bases are going to project inwards. The bases, they project inwards. Okay. So sugar and phosphate, they form the backbone of DNA, whereas the bases, they are going to project inwards. So in the double helix conformation, the backbone is formed by sugar and phosphate, whereas bases project inwards. Now how is the two double helix components held with each other? They are held together with the help of some bonds. One of the bonds is hydrogen bonds. Hydrogen bonds that form between bases in the process of base pairing. Now we had talked about bases in DNA, nitrogenous bases. These nitrogenous bases that are attached to the sugar with the help of glycosidic bond are of two types, purines and pyrimidines. Purines are more heavier as they are comprised of a 6-membered ring and a 5-membered ring, whereas pyrimidines are just comprised of a 6-membered ring. Now purines are of two types. whereas pyrimidines are of three types. Two types of purines are adenine and guanine. Whereas pyrimidines are of three types, we have cytosine, thymine, and uracil. Uracil base is only found in case of RNA, and thymine is not found in RNA. So in DNA will not have uracil, and in RNA will not have thymine. So how do these different bases they pair with each other? Always a purine will base pair with a pyrimidine. Okay. So adenine base pairs with thymine in case of DNA or uracil in case of RNA. They form two hydrogen bonds with each other and participate in base pairing. Now cytosine forms three hydrogen bonds with guanine. Now because three hydrogen bonds are involved here, it would take much more energy to break their bonds as compared to an AT or AU bond. So let's come back to the double helix structure. So in double helix, these hydrogen bonds that are formed as a result of base pairing help to keep the DNA more stable, right? So DNA exists in a double helix, and this double helix conformation is stabilized with the help of hydrogen bonds. The other bond that stabilizes DNA is the hydrophobic stacking of bases. So what basically is happening, all these bases, they are projected inwards and they are stacked one upon each other, and these bases are hydrophobic in nature. So when they interact with each other, that helps again to stabilize DNA. So for DNA to stabilize, two types of bonds are required. Hydrogen bonds that results from base pairing, and hydrophobic bonds that are formed due to stacking of nitrogenous bases. Both these bonds, they stabilize the secondary conformation of DNA. If I rotate the DNA strand by 360 degree, I'll find around 10 bases. So in a complete rotation of DNA strand, I find around 10 bases, right? Another way to say this, that the pitch of DNA, pitch simply refers to the rise per turn. So if I stretch this out in the form of a double helix, that would be called as pitch. So in a pitch, that is simply 360 degree. In a complete rotation of double helix, how many bases do I find? 10 bases. And what's the distance from here to here? It's around 3.4 nanometer. So distance between two bases is 0.34 nanometer. Okay. So the pitch of DNA is around 3.4. 3.4, that is, in a complete rotation, the length of DNA is around 3.4 nanometer. And in this complete rotation, I find about 10 bases. So that tells me that the distance between two nucleotides is 0.34 nanometer. So if we are given how many bases there are in DNA, bases or base pairs that are there in DNA, we can easily calculate the length of DNA by simply multiplying the number of base pairs into 0.34, that is the distance between two base pairs. Yeah. And the diameter of DNA is 2 nanometer. Now the DNA strands, they have a polarity. So at this end, I would have a free 3' group where hydroxyl group is there, and at another end, I would have a 5' end, right? So I can say in a strand of DNA, I have a 3' end at one end and a 5' end on another end. Using this, I'll be using it to define the polarity of DNA. So the two strands that are involved in double helix, they have anti-polarity. That is, they are in opposite conformation. They are anti-parallel to each other. So if one of the strands is 5' to 3', the other one would be 3' to 5'. So I would say that both these strands, they are anti-parallel, meaning that they have opposite polarity. If one is 5 to 3, other would be 3 to 5. Okay. And both these strands, they are complementary to each other. And this complementary feature is due to the fact that bases participate in base pairing, right? So each strand is complementary to the another. What that simply means is so I would say that if this strand is complementary to the other, then if here there is A, here there would be T. A, C, G, G, A, T, G. So you can say they are complementary to each other due to base pairing. So in organisms, we do find DNA as well as RNA as the genetic material. A genetic material, first of all, should be stable. It should be able to replicate itself. So there should be a mechanism of replication. And it should allow for changes to occur within itself. That is, it should allow mutation. So if we see the requirements of genetic material, both RNA and DNA are choice materials for genetic material. But even then, DNA is the most preferable genetic material. Why? Because DNA is more stable and less reactive. DNA is more stable due to the fact that it has hydrogen group at the 2' end, and second is the presence of thymine instead of uracil also confers stability to the DNA. Whereas RNA, being more reactive, and less stable, is more preferred as a messenger. Okay. So out of RNA and DNA, RNA might have been the first to evolve, and this hypothesis is known as RNA world hypothesis. RNA might have been the first genetic material to have evolved in the course of evolution, as RNA has both the properties. It can act like a genetic material and it can also act like a protein by catalyzing the reactions. It can also act like an enzyme and catalyze reactions, and it can also act as your genetic material. An example of an RNA that catalyzes reaction is ribosome. So due to the reasons that we have studied, DNA is the more genetic material, more preferred genetic material. Okay, so we are done with the structure of DNA. We have studied how the primary structure of DNA is composed of nucleotides that are joined to each other with the help of phosphodiester bond, whereas the secondary structure of DNA is formed by the double helix conformation. This double helix conformation was given by Watson and Crick. So as soon as Watson and Crick, they were devised the second double helix structure of DNA, what came to their mind is the mechanism of replication, right? Just by looking at the structure of DNA, they could say that there might be a way in which DNA replicates itself, right? We all know that our life starts from a zygote. From a zygote, a number of cells have to be formed, and in each of the cells, there has to be DNA present. So there might be a mechanism by which DNA is replicated in the course of cell division, right? So let's now look at replication. Okay. Now, before Watson and Crick gave their model of DNA replication, there were three proposed models of DNA replication. These were semi-conservative, conservative, and dispersive. Now, out of these three, the one that Watson and Crick gave was the semi-conservative model of DNA replication. Semi-conservative model of DNA replication. Now, what does the semi-conservative model of replication say? That because we have two strands of DNA which are antiparallel to each other, so when this particular DNA has to replicate, each strand is going to act as the template and they will help in synthesis of a daughter strand. So as a result of DNA replication, we are going to have parental strand, which was acting as the template strand, and the newly synthesized daughter strand. So this model is semi-conservative because here the parental strand is conserved in each round of replication. With the newly synthesized DNA, the parental strand is conserved. And this model is based on the complementarity of DNA. The new daughter strands that would be formed would be complementary to the parent strand. Right. And that is how they have also been synthesized. For example, in the parent strand there was ATGC, right? So opposite to A, in the daughter strand there would be T, right? So there would be complementary. Next, look at the experimental proof. So Messelson and Stahl experiment help to give the experimental proof of the fact that DNA is actually being replicated with the help of semi-conservative model. Okay, so in the Messelson and Stahl experiment, what they did is they took some bacteria, that is your E. coli, and they grew it in a medium that contained heavy nitrogen. N15 is the heavier isotope of nitrogen. The one that we normally have in our atmosphere is the lighter one, that is N14. Okay? So they grew bacteria for many generations in the N15 medium. So we just now studied that in DNA there are nitrogenous bases, right? So for forming these nitrogenous bases, DNA, or for making DNA, nitrogen is required, right? So the bacteria would be utilizing this nitrogen from their medium or from their surrounding for making DNA. So as more and more bacteria they replicate and grow, in all of them N15 would get incorporated. Right? It would get incorporated in them. Why? Because they are using it to synthesize their DNA. So in all of them there would be N15, right? Now after growing the bacteria for some generations in the N15 medium, they centrifuged it. Centrifugation is a process in which the materials or substances, they are separated out according to their densities. Okay? So the gradient they used was cesium chloride. So when in the cesium chloride gradient they performed centrifugation, they got a band. Now you have to remember that N15 was heavy. So we would get a band somewhere here. Being heavy, we would get a band at the bottom. Now what they did is after some time, after some generations, they transferred this bacteria into another medium. Now this medium contained only N14. Now N14 is the lighter isotope of N15. Now after growing the bacteria for some time in N14 medium, again they centrifuged it. Now on centrifugation, they got a band again. Now the band they got was lighter than the N15 band. They got a band here. This was the N14, N15 band. Okay, so let's understand what happened here. When the bacteria was growing in the N15 medium, it used N15 to form its DNA. So it's basically the DNA that is present here is your N15. And this DNA is heavy. Okay. After a few generations, when it was transferred to the N14 medium, there the bacteria already have your N15 in their DNA, right? So during replication, what will happen? Each of these strands is going to act as a template, right? And new daughter strands will be formed. Now the new daughter strands that will be formed will be using N14 that is there in their medium, right? So there would be different. So now the two strands that I'm getting as a result of replication, they are hybrids. Right? Why hybrids? Because one strand is N15 while the other one is N14 strand, right? So I'm getting a hybrid. So their weight would be somewhere intermediate to N14 and N15. That is the reason I'm getting a hybrid band here. I'm getting a hybrid N14, N15 band here. Okay. Now after a few more generations, this scientist again took some sample for centrifugation. Now what did they observe? They got another band that was lighter compared to the other two bands. So this is the N14 band. Okay. Again, what you can see is, after some time, after you grow the bacteria for many generations, what will happen is, so if suppose this is the particular strand of DNA, now the new DNA that would be synthesized would again be N14, right? So we would get an N14 band. No hybrid is there, N14, right? But here again this strand can also act as the template strand. So when this strand acts like a template strand, then we would get a hybrid. Then we are going to get a hybrid. So in the last one, we will get a lighter band and also an intermediate band, an intermediate hybrid band. Okay. So now, due to Messelson and Stahl experiment, they experimentally proved that the model that was given by Watson and Crick, that is semi-replication model, is the one that DNA replication follows. So this model was accepted. Okay. So now before moving on to the mechanism, how does DNA actually replicate, let's first understand central dogma. Okay. What does the molecular central dogma state? The central dogma states that from DNA, RNA is made, through the process of transcription. And from RNA, your proteins are synthesized with the help of the process translation. So from DNA, a messenger RNA is synthesized in the process of transcription, and from your RNA, proteins are finally made. Okay? So that is how we learned in genetics that the DNA was able to express themselves, right? What we saw in genetics is that a particular genotype was giving rise to a phenotype. And what is responsible for that phenotype? A particular protein, right? So how is a particular DNA able to express itself? A DNA expresses itself in the form of a protein that does some function to produce a phenotype. Okay. And we also studied that DNA can replicate, right? DNA can replicate. Okay. So this is known as the central dogma. Central dogma tells us from where to where information flows, right? It is telling us about the flow of information. So the flow of information is always from DNA to RNA to proteins. What about organisms where RNA is the genetic material? In their case, it would start from here, RNA to finally proteins, right? So in their case, the translation process is much more easier, and transcription is not involved in organisms who have RNA as their genetic material. So let's come to DNA replication. We are going to learn how does DNA replicate. So for DNA replication to occur, we know that DNA exists as a double helix strand, and DNA is a very, very long strand that has been packaged into the cells. Okay. So before DNA replication, let's also learn about DNA packaging. Now because DNA is such a long macromolecule, it is very, very lengthy, it is packaged within a cell. It is packaged within a cell. In the cell also within a nucleus it is packaged. So what helps DNA to package? There are certain proteins known as histones. Histones have high amount of arginine and lysine. What is arginine and lysine? They are basic amino acids. Histones, these proteins are made of large quantities of arginine and lysine, which are basic amino acids, and histones are positively charged. So that might give you a clue as to how histones are packaging DNA. DNA is negatively charged, whereas histones are positively charged, right? So they attract each other, due to which histones can easily package DNA. Okay. So let's see how histones are involved in packaging of DNA. Okay. Now histones form nucleosomes. What are nucleosomes? Nucleosomes are octamer of histones. So 8 histone molecules, they come together and form a nucleosome, and which all histones are involved in the formation of nucleosome? H2, H3 and H4, right? So these 3 are involved. H2A is there and H2B is there, right? So all these 4 types are involved in the formation of nucleosomes. And we have 2 of each. Okay. So all these 4 types of histones, they come together and form nucleosome. Now this nucleosome is again positively charged. Now to this nucleosome, DNA is tightly packaged. Now to these nucleosomes, DNA is tightly wound. There is 2 turns of DNA per nucleosomes. Right. So if this was one nucleosome, we would have another nucleosome here. And this piece of DNA that connects one nucleosome to the other is known as linker DNA. And in this linker DNA, there is another histone molecule present that is not directly involved in packaging, right? This is H1. This is the histone H1. Here, histone H1 is present. So, histone H1 is present in the linker DNA, whereas the 4 different histones form octamer in the form of nucleosomes. Okay? So, histones are involved in packaging of the DNA. If this is a nucleosome, 2 rounds of DNA are packaged. Right? And how they are packaged? Due to their opposite charge, they hold onto the DNA. So, this packaging of DNA is seen in case of eukaryotes. Only eukaryotes have histones, whereas in case of prokaryotes, there are other sort of proteins that are involved that help to package DNA, but there the packaging is not as detailed, not as organized as in eukaryotic cells. Okay? Now, histones help to package DNA, right, and they give the DNA a beads on string model. So, if you see chromatin, it would be visible something like a beads on string model, right? But we know that the chromatin can be further condensed. In metaphase, we see not chromatin but rather chromosomes. In chromosomes, the chromatin is highly condensed. So, for this condensed form, DNA needs to be more packaged. So, that tells us DNA can be more packaged, more tightly packaged, right? For more tightly packaging of DNA, besides histone, we also require non-histone proteins. Okay? Certain non-histone proteins are also required for the further packaging of DNA. Now, this is a nucleosome, and I just told you that there are 2 turns of DNA per nucleosome that are tightly bound to the nucleosome. Right? Now, if we observe the strand of DNA very closely, we find certain strands of DNA, they are very tightly bound to the nucleosomes, that is, they have high affinity for the nucleosomes, right? So, in such conditions, what would happen? There would be compact packaging. So, in compact packaging, where the DNA is packaged very tightly, the DNA is not free. Now, because DNA is not free, it is not able to express itself. So, here transcription and translation won't be occurring. So, such type of DNA which is inactive in the process of expressing itself is known as heterochromatin. Okay. So, heterochromatin is a segment of DNA that is packaged very tightly and they are not able to express themselves. Okay. Now, the affinity of DNA and histones, they can be modified. For example, if this is the nucleosomes, by adding certain tags, if we add certain tags like methyl, acetyl group, on adding these tags, the affinity of nucleosome and DNA can be altered, right? So, in some parts of DNA, we have certain regions where the DNA is very loosely packaged, where the DNA is not very tightly packed, but the DNA exists in a loose conformation. So, here what we see is DNA is loosely packed. So, the DNA is free to express itself. So, here translation, transcription, everything would be occurring. So, such type of DNA that is not packed very tightly and can express itself is known as euchromatin. Okay. So, in our DNA there are regions of heterochromatin and then there are regions of euchromatin. Euchromatin are basically those strands of DNA that keep on expressing themselves, right? More genes that are always active would be present on your DNA, on your euchromatin. Okay. So, let's come back to DNA replication.